Hello, everybody. Today, our show will be talking about DNA, protein synthesis, and protein functions. Stay tuned. Welcome to The Voice of Sparta. I'm your host, Isaias, and I'm joined by a special guest.
Hey.
So, Edwin, it is my pleasure to welcome you to this podcast. I've heard that you might have some questions related to DNA,
Yep.
and I'm here to help. So, what questions do you have about DNA?
First of all, I've had a difficult time understanding the structure of DNA.
The structure of DNA. DNA is a double helix shape, but you probably already know that.
I understand that part, but my question is, what is DNA made out of?
So inside DNA, there are these three components called nucleotides. These nucleotides are made of three subunits: the phosphate, the deoxyribose sugar, and the nitrogenous base.
What's a nitrogenous base?
A nitrogenous base is the building block of DNA. There are four possible bases: adenine, thymine, cytosine, and guanine.
What do these four bases do for DNA?
These four bases are in pairs and are complementary to each other. For example, an adenine base would pair up with a thymine base, and a cytosine base would pair up with a guanine base. This rule essentially creates many combinations of these two pairs.
Okay. But do these pairs stay connected without being ripped apart?
As I said, DNA is a double helix, meaning that there are opposite strands to this shape. One combination of bases runs along one side and the complementary combination runs on the other. When these bases connect, they are held together by hydrogen bonds in the middle.
Okay, I'm starting to understand DNA a bit better. However, in my notes, I have DNA backbone written. Can you explain that to me?
The DNA backbone, also referred to as the sugar phosphate backbone, is like the name implies. It is the backbone of the nucleotides. It is what keeps them held together across the DNA strand, thousands at a time.
Okay, I think I'm getting the hang of it. Now that we're here, can we talk about protein synthesis?
Of course. What do you know about protein synthesis?
I know that protein synthesis starts with transcription, more specifically with the splitting of DNA by our mRNA.
Correct?
Then mRNA transcribes the DNA genetic code into its own, but replaces thymine with uracil.
Yup. And then translation follows. mRNA takes a transcribed code and goes to the ribosome.
And in the ribosome, mRNA is translated to tRNA. The mRNA code is read in triplet pairs called codon triplets.
Then tRNA brings amino acids according to the codon code, with the help of anticodons.
And there are 21 different amino acids, all of which have different shapes and sizes. Put together, these amino acids create a singular protein.
This chain of amino acids is called a polypeptide and is the primary structure of a protein. Did you know that protein shapes is affected by the amino acids?
I do. Since the amino acids are made of different atoms, putting them different amino acids together would mean that the polypeptide backbone would rearrange into a different shape every time. This affects the secondary structure and the tertiary structure.
The secondary structure can be composed of an alpha helix or a beta sheath, and many of these two shapes create a 3D structure referred to as a tertiary structure. This results in the final shape of a protein.
I know that the structure of a protein affects its function. For example, hemoglobin is a protein that helps keep oxygen molecules and iron at its inside, away from outside molecules. Its chubby shape allows it to execute this job.
Yes, sir. Hemoglobin is crucial for the circulatory system. Without it, red blood cells would not work properly and you would not receive oxygen throughout the body. So think about that every time you go out for a run. Do you have any other examples you would like to share?
I can only think of antibodies, but I'm unsure of what their structure is like.
Antibodies are shaped with multiple long arms. This helps it bind to multiple foreign invaders at once, eliminating them with ease and efficiency. Without these antibodies, our immune system will be weak to disease and could be fatal.
And all these processes of creating a protein are affected by the structure of DNA?
Yes, because DNA sequences of nucleotides are transcribed and translated, those same codes would affect the types of amino acids chosen for the protein.
And those amino acids would affect the protein shape because of the different structures of amino acids. Finally, the shape would help proteins with their job as different shapes provide different advantages. Tracing back these steps, we can see the DNA structure affects protein shape and its function.
This is why DNA is considered the building block of life. It allows our body to function properly, supporting us from birth to death. Any mishap could create an undesirable protein, and we are prone to these consequences.
Thank you for helping me understand DNA and protein synthesis better. I tried to gain information from other sources, but I must say your explanation was much easier to interpret.
Anytime. Thank you for coming to talk on the podcast, and thanks to the viewers for listening.